Oh. Hmm. स्लाइड्स नहीं आई हैं यार। एक मिनट रुको, अभी मत करना। स्लाइड लोड नहीं हुआ है, चलो तो इसीलिए लैपटॉप से फिर से जाना पड़ेगा। लैपटॉप में हाँ अभी आया। एक इशू है, स्लाइड लोड नहीं हो रहा है, जैसे अभी आया था ना, अभी फिर से मैंने देखा तो वो गायब हो जा रहा है बीच का। समझा? देखो यहाँ पे कुछ था, गायब हो चुका है। रुको बाकी एक बार देखने दो पहले, ये सब गायब हो गए। अभी था, वही समझ नहीं आ रहा। स्लाइड फाइव में क्या था? स्लाइड फाइव में क्या था? स्लाइड फाइव में इमेज था एक, सेम फॉर अदर स्लाइड्स। ये भी कट के आ रहा है। हाँ नहीं ठीक है, ठीक है, लेकिन बाकी भी हैं। 8 9 10 सब गायब है। तो पहली बार वही मैं सोच रहा हूँ। 5 6 7 8 9 10 गायब होगा, फिर 11 कुछ नहीं हो रहा है। क्या 8 से वापस? अरे भाई प्रॉब्लम इसकी वो दिखा था जब पहली बार तुमने करा था, और उसके बाद वो गायब हो गया। ये कैसे हो रहा है? हाँ बहुत अफसोस है। हाँ देख रहे हो। एक काम करो क्या? पेट पड़ा उसके बाद वापस इतना तो कर ही सकते हो। वापस मतलब? ओ से 8 तक पढ़ाओ, हाँ उसके बाद वापस कर सकता हूँ, चेक मेरे स्लाइड बोलो। अरे यार वे तो मैं बार बार करना पड़ेगा ना, और डिस्टर्ब होगा बहुत। 9 से ट्राई करो, 9 से 13 तक डालते हैं। 9 से अगला अगला स्लाइड है, एम्प्टी वाला 9 बोलो। अच्छा हाँ इधर से करो। हाँ 9 से करो। 9 से कितना था? 13 शायद। हाँ हाँ अभी चेक करो। 9 क्या प्रॉब्लम है इसका? ट्राई कर सकते हो, थोड़ा वो हो जाएगा ना। एक बार पूरा हटा दो, एंड कर दो क्लास। हाँ सेंड करके फिर नहीं किसी और के में लॉगिन करते हैं, त्रिभुवन सर के में दिक्कत नहीं आती ना? नहीं देखो इसके एक ही सॉल्यूशन मैं सोचता हूँ जो हो सकता है। क्या? टेलीग्राम, टेलीग्राम में नोट किया था तुमने वाला स्लाइड? हाँ बोलो वो ये है। ठीक है फिर इसका ऐसे ईमेल अगर पीपीटी भाई ये पहली बार हो रहा है, इसके पहले कभी नहीं हुआ। वही तो मैं सोच रहा हूँ। डाउनलोड्स में है वो। ठीक है हाँ पर उसमें लिखेंगे कैसे? ओ ये तो फिर ये नेक्स्ट पे कैसे जाएगा? ऐसा भी कर सकता हूँ। अरे यार ऐसे करना पड़ेगा। हाँ, यही ऑप्शन है। नहीं बट रुको रुको, नेक्स्ट पे कैसे जाएगा? नहीं 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 नहीं, नहीं, ऐसा करो, इससे तो मैनेज करोगे। हाँ नेक्स्ट पे कैसे जाओगे? तो मैं कह रहा हूँ इससे नहीं होगा, ये तो बेकार है और भी तो फिर आई थिंक रुको उसी से करते हैं, बट उससे ऐसा नहीं होना चाहिए। अच्छा, क्या? समझ गए क्या जा वापस जाओ। हाँ। जब तुम इस पे एडिट दबाओगे, फॉर एग्जांपल दिस, तो तुम्हारा ये खोलेगा। ठीक है, ये तुमने लिख दिया, कर लिया। हाँ। अब ये
नेक्स्ट
तुम
पेज
करोगे कंफर्म, नेक्स्ट पेज
ऐसे करना पड़ेगा।
ऐसे ही करना।
ठीक है
बट
hello everyone. So sorry for the delay. There was some problem in the loading of slides, but we will manage with this today. Okay. So today we will start the first lecture of cell, the unit of life, right? So in the previous one, लेकिन एक इशू और भी तो है, ये नहीं वर्क करेगा। कैसे करना पड़ेगा? इसका इशू है। रंजीत उधर आओ। उस पे मैं स्लाइड ऐड नहीं कर सकता हूँ ना, मुझे स्लाइड वही दिक्कत है, उसी से ही करना पड़ेगा। अरे यार हम्म, ऐसा क्यों हो रहा है, मुझे समझ नहीं आ रहा।
पहले क्लास करोगे ऐसा तो नहीं कि बहुत बड़ा वीडियो।
तो इतना तो इसको ले लेना चाहिए यार,
हम लोग
लोग
को पहले इसमें यूज किया था ना।
वही तो, लोग तो कितना पढ़ाते हैं।
क्लासरूम का तो चाहिए था।
या तो उसको थोड़ा थोड़ा करके इम्पोर्ट करूँ। um, these, करो यार, ऐसा इशू नहीं आता कभी। हम्म, इसी में से कुछ सिलेक्ट कर लो।
करो।
नोट्स।
ये मैं बोलूंगा ना, चेक मत करो, नेक्स्ट टाइम अगर एक भी स्लाइड आ गया ना पढ़ाना तो बंद हो गया तो फिर देख लेना।
हाँ, बड़ा दिक्कत है यार, ऐसे क्यों हो रहा है समझ ही नहीं आ रहा।
इस बार ना सारे स्लाइड्स आए थे ना,
स्लाइड्स पहले आ रहे हैं फिर वो वापस लोड नहीं हो रहे
वापस चेक करके देखना है तो
हैं, तो पहली बार में पढ़ाना शुरू कर दो।
पहली बार पढ़ा
वो भी कर सकते हैं।
देख लो।
ठीक है। Okay, so let's start with the class. Okay, so today we will start the first lecture for the cell, the unit of life, right? So I hope you all know about the basic of membranes, right? That we have discussed, right? How a monolayer is formed, how a bilayer of membrane is formed. So if we talk about a cell, okay, so what do you understand by the term cell? Anyone?
The most basic structural and functional unit of
Structural and functional. Are you missing some point?
fundamental.
Fundamental unit of life, right? So we define a cell simply as the structural, fundamental, and functional unit of life, right? So the meaning of the statement is very simple, that it is the structural unit of life. That means that the structure of all the living organisms which is there, okay? So if you consider yourself, you are made up of these cells, right? So there is a structural unit that is combining itself and is forming an organism which is functional, which is able to perform some functions, right? And also it is the fundamental unit of life. That means that nothing smaller than the cell can actually exist for a living organism if you consider, right? So this was about a cell. Now, according to NCRT, if we see what is a cell, so NCRT, it tells us that unicellular organisms are capable of independent existence, right? So that means that these organisms, they can exist independently, right? They are not dependent on other organisms. For a particular cell, if you understand a cell in very simple terms, you can call it as a compartment in layman terms, right? So a compartment means that it surrounds the inner of the material and everything from the outer environment, right? So inside this compartment, a lot of activities are undergoing, a lot of chemical reactions are happening, and this is able to sustain itself independently. Living actually, what organisms you can classify as living or non-living?
Consciousness and the ability of cellular.
Correct. So for living, we define this in few terms, like one is consciousness, right? And cellular organization, correct? So consciousness means the ability to respond to the external stimuli, right? So basically for this, you require the cells. And now if we talk about what the cell is made up of, right? So as we have discussed, if you talk about this, so during the formation of the, you can say when the earth was forming, okay, so what happened was in the primordial earth, somewhat like billions of years ago, what happened was there was these molecules, okay, which we call as lipids, right? And now these lipids which we talked about, these lipids can be of two types, right? First is the asymmetric lipids, and the second one can be symmetric lipids. So, can anyone tell what are asymmetric lipids? Okay, so for asymmetric lipids, you have a head group that is there, but a single tail, right? And for symmetric lipids, what happens is you have two tails, which these tails are what? Can anyone tell? What are these tails? Hydrophobic, but what they are made up of? Long chain carboxylic acids, right? Which we call as fatty acids. So basically, if you visualize this structure, this appears as a cylindrical structure which is symmetric because the head group is proportionate with the tail, right? So overall this is a symmetric structure, but if you consider this structure, this you can visualize as a cone, right, which is asymmetric. So now there are different possibilities if you add this lipid to water, okay? So there are different possibilities, and these possibilities basically they depend on the concentration of the lipids that you are adding, right? So one can be the possibility of formation of a monolayer of lipid, and the another one can be the formation of a micelle. So can anyone tell when we have a monolayer and when we have a micelle which is formed? Below critical micelle concentration, when your concentration of lipids is less than the CMC, you get this. And when the concentration of lipids is more than CMC, you get micelle formation. Now, how does a micelle look like? So, if you are surrounded with water, first thing you need to understand is this head group is hydrophilic. That means it is water loving, right? And the tail group is hydrophobic. That means it is water repelling, correct? So, now what happens is for the micelle, if everywhere you have water, right, what will happen is this head group will be facing towards the water, and the tail groups they will form the core of this particle, okay, which we called as a micelle, right? So I hope this part is clear to everyone, correct? Now if we talk about symmetric lipids, so for symmetric lipids, if you add these lipids in water, basically what happens is, again, you can see the formation of a bilayer of lipids, or you can get the formation of a liposomes, right? So how does a bilayer formation looks like? So when you have these lipids, they are arranged in such a manner that they are forming a two layers. This is the first layer, this is the second layer, right? And if the you can say the space allows, and if you consider here everywhere there is water, right? So under favorable conditions, this bilayer can fold and it can basically form a structure called as your liposomes, in which what happens is they can act as a delivery vehicle, right? And in them, basically in the core part, you have the lipids which are oriented like this, and here the lipids are oriented towards water. So this is how the liposome formation happens for the symmetric lipids. Now considering the cell, so now we will understand in detail how these lipids actually look like, and also how you can say the structure of lipids is there, right? So first thing is, as you have noticed, we have a head group and we have a tail, right? So for the head group is hydrophilic. Now why it is hydrophilic? So to understand this part, first we will understand about the formation of the lipids for your cell membrane, right? So if I draw one structure, can anyone tell what this structure is? This is glycerol, right? How many OH group it has? 3 OH groups. Now, we talked about the glycerol. Now we are talking about the tail part. Okay, so for the tail, they are long chain carboxylic acids, which we also called as fatty acids. So there are a lot of fatty acids, but for our simplicity, we will draw them something like this, where you will have mainly this kind of groups, right? And let's say you have the 3 of the fatty acids. Okay. So these are carboxylic acids, long chain carboxylic acid. So this can repeat n number of times depending on the type of fatty acid you are talking about. And what happens is, if you consider this group, okay, so what will happen is basically the hydrogen, and here what will happen is, here you will have the OH, right? So these will react, okay, and this process is called as esterification, where you have a formation of esters, okay. So overall you will get some structure which is like this. So if you have ever seen a structure of ester, then you will be able to appreciate the structure. And so if you consider overall this structure, right? So ester is something like this: RC double bond O R dash, right? So here you have the ester linkage which is present here also, right? So this is the structure which is formed by the reaction of glycerol with fatty acid, okay? And this is how you have formed these lipids. This is the lipid structure, right? Now we will understand this structure in this way. So what is happening is, if you see the structure here, you have the glycerol backbone, right? And you have the basically the reacted groups, okay? So this is the continuation of that. But here you can see on one of the CH2 you have a phosphate group attached, right? Now this phosphate group is the reason that this head group is hydrophilic in nature, right? Because it has a negative charge, correct? And now if you see the tail, so the tail, it can either be made up of saturated fatty acids. What are saturated fatty acids? Saturated fatty acids which have single bonds, correct? And unsaturated fatty acids which have double bonds, or you can say triple bonds, right? That is unsaturation. So if you consider overall the structure, this is how it is made up of. So basically here you have the head group, right, which has the phosphate and the glycerol backbone which is there, okay. And if you see the structure, basically here in this place, instead of having this basically this backbone of the fatty acid, here you simply have the phosphate, okay. So that is how you can understand about these lipids. Now this was the basic architecture. Now depending on the type of backbone that we have, so this was one example of the lipids which are found in our plasma membrane. But depending on different backbones, you can have different classes of lipids, okay. So the first one, if you consider, this is the one which we studied, okay. So basically you can have glycerolipids, you can have phospholipids, and you can also have sphingolipids, okay. Now don't worry about the name of all these, you don't have to exactly remember them. This is there so that you can understand that why the lipid membrane is asymmetric. So we will come to that point, but for now let me briefly tell you about this. So in the glycerolipids, you can have triacyl glycerols, okay, triacyl glycerols. In these, what happens is, you will have a glycerol backbone, right? You will have a glycerol backbone. So this backbone which you saw here, this is the glycerol backbone, right? You will have this backbone. Apart from the backbone, you will have 3 fatty acids which are linked, right? The structure which I drawed earlier. So these are triacyl glycerols. They are found in your adipose tissue and your blood lipoproteins. Now this is the most important category, that is your glycerophospholipids. So in glycerophospholipids, the backbone will be of glycerol only, but 2 fatty acids will be there, and in the third carbon you will have the linkage of the phosphate, okay. Now this is a phosphate. To that phosphate there can be attachment of a head group, okay. Now what is that head group? That head group, there are different head groups like choline, ethanolamine, inositol phosphate, glycerol, okay, and cardiolipin. So you can have different groups which can be attached to this structure. Depending on that, you will have different phospholipids, okay. You abbreviate them as phosphatidyl choline, phosphatidyl ethanolamine, phosphatidyl anisotol phosphate, okay, and there can be different variations. It can be PIP2, that is bisphosphate, or PIP3 also, okay. Like that, you can have different inositol phosphates and phosphatidyl glycerol. All these are the different variations of this structure, okay. No need to remember these, you just have to briefly understand that depending on the structure, you can have different, you can say, lipids, okay. Now, talking about ether glycerolipids. So ether group is simply RO R dash, right? I hope you know about this. So in this case, you will have a glycerol which is linked to a ether linkage. Okay, with this linkage, will have a ether. In the second carbon, you will have fatty acid. In the third carbon, a phosphate and a head group. Okay, so these are basically present in the platelet activating factors. Okay, so when you get a cut in your body, then what happens is there are a lot of cascade of proteins which acts so that the clot can form, right? So for them, these lipids are involved in those factors. Okay, now there is another category, separate category of sphingolipids. Okay, so these sphingolipids, they are again of two types. One is sphingophospholipid. Okay. So in this, you have sphingomyelin. Sphingomyelin is found in neurons. Okay. In neurons. So if you know about the structure of neuron, what happens is you have a structure like this, right? So in this, basically, if you see the axon and the dendrite, so sphingomyelin is something which is present in these membranes. Okay. So in this, what happens is the backbone is no longer of glycerol. The backbone has now been replaced with sphingosine. Okay. Sphingosine is a chemical structure. No need to go into the detail. And then you have a fatty acid, phosphate group, and the head group. Okay. So this is sphingomyelin. Now talking about the glycolipids, okay. So in this you will have different categories like gangliosides and cerebrosides and like this. So in this, attached to the phosphate backbone will be sphingosine, but you will have a sugar moiety that can be attached. So basically for you, this is important to know, okay, and these are very important to know, okay. So now if you consider all of this, what we need to understand is that your plasma membrane, okay, your plasma membrane, it is rich in the majority of the plasma membrane, the lipids which are present in the plasma membrane, majority of them are glycerophospholipids. And among the glycerophospholipids, the lipid which is found in the majority, so you can see in this category, you have a lot of lipids, right? You have a lot of lipids. Among them, phosphatidylcholine, which is also, you can call it as lecithin, this is present in the maximum amount. Okay. So among this, lecithin or phosphatidylcholine, this is present in maximum amount. So this is a question that can come in your NEET or mostly NEET kind of question exam, that in the plasma membrane, which lipids are found in the maximum amount, right? So if you have glycerophospholipids, sphingolipids, all these options, you will mark glycerophospholipids. If you have option as phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine, then you will choose phosphatidylethanolamine, or if the name lecithin is given, then you will write mark that. Is it clear to everyone, this part? Okay. Now, since you have these different kind of lipids, what happens is in the plasma membrane, okay, in the structure of the plasma membrane, what happens is, so this part is called as the outer leaflet of the membrane, and this part is called as the inner leaflet of the membrane, right? So if you consider this particular thing here, what happens is in the outer leaflet, so how we are classifying it as leaflet, so you can simply imagine like this. So if you have a leaf, okay, you watch it from the top, okay, so you will call this surface as the outer leaflet. And if you are watching from here, you will call this one will be inner leaflet, okay. Like that, if you are watching from here, this part which is towards the surrounding, okay, which is so here you have the outer environment. So this will be called as outer leaflet, and here you have the cytoplasm, right, of the cell. So if this is the membrane, this is the cytoplasm of the cell. So this one is the inner leaflet, okay, which is towards the cytoplasm. Okay. So what happens is the amount of lipids in the outer and the inner leaflet, they are different. Okay. What happens is the phosphatidylcholine or the sphingomyelin, these lipids basically they are present at the outer leaflet, and the remaining ones are mostly in the inner leaflet. So the distribution of lipids is also different in case of your cell membrane. It is not that in the outer and inner leaflet all these lipids will be present in exactly same proportion. They are present at different amounts, and this is something which leads to, this is one of the reason which leads to the transverse asymmetry of plasma membrane. Now, if someone asks you, is the plasma membrane symmetric or asymmetric? So the answer is it is asymmetric. Why? Because the distribution of lipids in the outer and the inner leaflet is different. And also there is one another reason that we will see in the next slides. Okay. But for now, you need to remember that plasma membrane, okay, for your level, you have to remember it is asymmetric. It is the majority of the lipids which are there are glycerophospholipids. Among glycerophospholipids, you have phosphatidylcholine, which is present in the maximum amount, and also that is also called as lecithin. So this is important for you. And if you talk about the architecture of glycerophospholipids, then you will have a glycerol backbone. You will have two fatty acids. To the third carbon, you will have a phosphate group which is attached to any head group. And in this case, you will only have, basically you can have these different head groups: choline, ethanolamine, inositol phosphate, like that. So this is about the membrane part. Is it clear to everyone? Any doubt? Clear? Yeah. Transverse asymmetry means, like, what happens is if you consider these two leaflets, right? So these are not symmetric. We are talking in terms of these. Okay. So what happens is in the outer leaflet, let's say you will have, so if you draw it in form of a graph, you will see that these are the levels. So the phosphatidylcholine here is more, the sphingomyelin here is more, but the other lipids, right, which are phosphatidyl ethanolamine, okay, and other lipids, they are present more in the inner leaflet, because of which asymmetry arises in the lipid composition in both the outer and the inner leaflet. So that is why you have this transverse asymmetry. Okay. One more reason is there for asymmetry that we will discuss. Now, coming over to the factors which affect the fluidity of the membrane. So this part is very important. Okay, it is important for IIT, okay, and also for NEET and NEST also. In all the three, you can have questions from this part. Okay, now let's understand about the fluidity of membrane. Before going into that, let's understand it from a basic term. Okay, so now what is fluidity? How do you define fluidity? Ability to flow, right? So what happens is if you consider fluidity in case of membranes, right, what it means is the membrane which is there, okay, the membrane which is there, the membrane can move, okay, basically by the movement of membrane we means these lipids, okay, the lipids which we are talking about, these lipids can move from here to here, okay, or this lipid can come from here to here. This is called as lateral movement. Okay, lateral movement means within, either in left or right, like that, you can understand. So this is the lateral movement of the lipids. Now, so if you understand this, that the lipids can move, that means the membrane is fluidic in nature, right? The membrane is fluidic in nature. That means the membrane is not a rigid structure. It is not something like a solid ball which cannot be, you can say, changed. You cannot change its conformation, right? It is not like a solid ball. It is fluidic in nature, right? So the membrane can have invaginations, the membrane can grow itself, the membrane can fold itself. It is fluidic in nature, okay? And this is the same thing that you study in the Singer and Nicholson model, okay, of the plasma membrane, that the membrane has a quasi-fluid nature. The membrane is not a rigid structure, it is quasi-fluid. That means it can change its shape, it can change its orientation, it can form different structures. That is all because the membrane is fluidic, right? Now the factors which affect this fluidity, we have to consider those factors. So factors affecting fluidity of membrane, okay? So if you consider those factors, the first factor will be the presence or absence of the, you can say, fatty acids. So if you talk about the fatty acids, they are of two types. One is unsaturated and the another is saturated. So now what happens in both the cases, right? So if you have unsaturated fatty acids, so what does it mean? You will have double bonds between carbon or carbon, right? So what will happen because of the double bonds? So if you consider a membrane which has these tails, right? So whenever you have a double bond, what happens is the orientation changes, okay? So what happens is, if anywhere you find a double bond, the lipid architecture will change. Basically, if the lipids has to be like this, if the packaging was like this, now what will happen is the architecture of the lipid has changed, because overall, if you compare this with this structure, so you can see this structure is highly packed, right? This structure is highly packed, correct? All the lipids are in the orientation which is required, like it is very packed structure, okay? No deviations, okay? No, you can say disformations, but here you can see because of a double bond, okay, what happens is, if this was the structure going, then suddenly the structure can change because of the double bonds. Because these double bonds, they introduce kinks. Kinks means some kind of gaps, you can say, in the membrane, okay? So because of which, what will happen is the fluidity, okay, the fluidity will increase. Why the fluidity will increase? Because the packaging is disturbed, okay? The packaging is disturbed. Here the fluidity was very good, this lipid can come here, this lipid can come here, things can be transported. But now as you have an unsaturated tail, this will change this packaging, okay? This will make some gap, this will change the packaging, and because of which the fluidity will increase. So you can see the case of unsaturated hydrocarbons, what is happening is, these tails, they are forming the double bonds, right? Because of which what is happening is that you are getting kinks and the packaging is being disturbed, because of which the fluidity has now increased. Now the membrane has more gaps, now the membrane can flow more in that particular area. But in this case, if you see, when you have the um, saturated fatty acids, right, then the fluidity, what will happen to the fluidity? It will decrease, correct? The fluidity will decrease because this is highly packed structure. This is highly packed structure, okay? So because of which the fluidity will decrease if you have the saturated carbon. Now this decrease means it is in comparison to the unsaturated, okay? These are relative terms, okay, not absolute terms, these are relative terms. With respect to unsaturated, this has less fluidity, and this unsaturated fatty acids will have more fluidity, correct? Now coming over to another factor, that is temperature. So if you talk about temperature, what will happen if you increase the temperature? What will happen if you decrease the temperature? If you increase the temperature, what happens is the kinetic energy increases, the molecules vibrate more. Then what will happen is, if the temperature is increased, the fluidity will increase. Okay. and if temperature decreases, the fluidity decreases. Correct? So this is another factor about temperature, how it affects the fluidity. Is this part clear to everyone till now? Right? Now there will be another factor which is nothing but cholesterol. Okay. So the presence of cholesterol, it also affects the fluidity. Now this cholesterol is present in the cell membrane only. Okay. And how does the cholesterol looks like? So if you see this membrane, these are your basically head group, tail group, and these are the cholesterol molecules. Okay, these are the cholesterol molecules. Now if you see, what happens is, this is the structure of cholesterol. So cholesterol, if it has to be present in this core, so this part is hydrophobic, right? So if the cholesterol has to be present here, it should have a non-polar hydrophobic tail kind of a structure, right? So that it can be inserted inside the membrane in this part, right? Now, if you talk about the rings of the cholesterol, okay, so what happens is, the hydroxyl group, it makes the head polar, okay, and that's how it can interact with the, you can say, the hydrophilic head of the lipids which are present in the membrane, and then this is the ring structure. Now, how cholesterol affects fluidity? So there are two cases. One is at moderate or higher temperatures, and the second case is at colder temperatures. So what happens is, if you have very high temperature, you imagine, if you have very high temperature, all the lipids, they are moving in a very fast pace, okay, the motion is very fast. So what happens is, in high temperature, the fluidity is already high, right? Now if you introduce cholesterol molecules in between, so these will do what? These will basically hinder the movement of the lipids, okay, because now you have inserted the cholesterol in between, right? In this case, what happens is that the fluidity which was high, it decreases. So in this case, the fluidity decreases when you talk about higher temperatures. Correct? Now you imagine a scenario of colder temperature. So when the temperature is cold, right, the lipid, basically you can say, they are tightly packed, okay, they are not moving much, right? So they are very tightly packed. And now if you suddenly introduce a cholesterol, this will disrupt this packaging, correct? Once the packaging is disrupted, the lipids will start to flow a bit. The flow will increase, right? So at colder temperature, what happens is, if you introduce cholesterol, this will increase the fluidity. Is this part clear to everyone? Any doubt? Yeah. Uh, okay. You can close that. So is this clear to everyone? Any doubt? Okay, see, you imagine it like this. So let's say all the students in the class, they are running here and there, right? They are at a high temperature, they are running here and there, they are colliding. The movement is very fast, the fluidity is very fast. Now what will happen is, if suddenly few of the bouncers came and they say stop, okay, so they stopped you, right? So then suddenly when the bouncers came, they decrease the fluidity, right? Because they have stopped you. Now you imagine each one of you is present in a line, okay, in a continuous line, okay, and now if suddenly someone came to fight you, he just throwing each one of you here and there, right? So he's fighting with you, he is just disrupting the line. So in that case, the fluidity increased, because initially you were tightly packed. Now once the bouncer or someone came, he is just punching you, he is throwing you away. So in that case, the fluidity increased. But in the previous case, you were already under high motion, you were running here and there. In that case, once you were hindered by someone, then the fluidity dropped as respect to the initial value. Is that clear? Clear to everyone? Okay. So if this is clear, then we will study about the in detail structure of plasma membrane. Okay. So this is how the membrane looks. Okay. So you have a lot of things. So initially what we have drawn were the lipids, right? Initially we have only drawn the lipids, but now you will see there are a lot of things in the membrane. So basically you have these peripheral proteins, which are some proteins which are interacting with the lipid head group. Okay, why the proteins are able to interact with the head group? Because the proteins, they are made up of amino acids, and in the elementary class you have understood about positively charged, negatively charged amino acids, correct? So with that, basically these proteins, they can be positive or negatively charged, and they can interact with this you can say hydrophilic part of the basically the membrane lipid. Now, attached to the proteins, there can be some other proteins, right, which you call as glycoproteins. Now, the meaning of the term glyco is sugar. The meaning of the term glyco is sugar. Okay. So basically these proteins which have the sugar moieties attached to the proteins, they can also be present on the membrane. Okay. Now talking about the glycolipid. Okay. So what can happen is that some of the sugar moieties, they can directly interact with the lipid part. Okay, that those lipids are basically, you can call them as glycolipids, because these carbohydrate moieties, they are interacting with the lipid. Okay, so they are called as glycolipid. Now another thing is, proteins can be of two types. One is the protein which is present on the periphery. So now if you understand this layer, this is the outer leaflet, this is the inner leaflet, right? So now if you consider here, there you will see the proteins which are either present on the periphery, only on the surface, and the proteins which are present throughout the membrane, that is they are also interacting with the outer leaflet, also with the inner leaflet. Those proteins, they are called as transmembrane proteins, or they are called as integral proteins. Okay, they are called as either transmembrane or integral proteins. These integral proteins, they can either be fully embedded inside the membrane, or they can be partially embedded in only one of the leaflet. For example, in the outer leaflet, you can see this protein, right? So this can be possible. Now, apart from that, you have cholesterol, which also act as membrane stabilizers, as I told you. And at this case that you have seen previously at colder temperature. So this is present in some of the, you can say, fishes in the Arctic region. Okay. So there what happens is, at colder temperature, these fishes, their cells, basically the membrane fluidity is very less. At that point of time, cholesterol helps in increasing the fluidity of the membrane. So they have inherent mechanisms by which they can increase the synthesis of cholesterol. Once the synthesis of cholesterol increases, the membrane fluidity is maintained. So that's why we called cholesterol as a membrane stabilizer, because it maintains the fluidity of the membrane. Okay, so this was one point. Now if you talk about the movement of lipids, okay, so basically this two monolayers, they are forming the bilayer, right? And if you consider the lateral movement of lipids, basically lateral movement can happen for both lipids as well as proteins. What does this statement means? So if you consider, in this case, if you have the lipid, right? So for the lipids, it is possible that the lipids, if you mark this lipid, okay, then it is possible that this lipid can move so this lipid can move here and this can come here. So this is something we call as the basically the lateral movement. Okay, so that means that the lipid can move within the leaflet, that is lateral movement. And if there are some proteins also which are present and they are attached to the lipid, these proteins can also move along with the lipid. So that is why the lateral movement, this is a very important question for your statements. The lateral movement, okay, this lateral movement, it is for both, it is shown by both, or it is present in both lipids as well as proteins. Okay, this is present in both lipids as well as proteins, correct? Now If you think about another kind of movement that you called as a flip flop movement, okay, so by the the word also you can understand flip flop. That means this lipid it can come to this leaflet and this lipid can go there, right? So this is flip flop movement. Now if you consider the flip flop movement, okay, if you consider the flip flop movement, right, so what will happen is this lipid will go to the inner leaflet and this one has moved to the outer leaflet. So in this case what happened is, you can see this is called as a flip flop movement, which is between two of the monolayers, right, which is between two of the monolayers between outer and inner. So this is called as flip flop movement. And flip flop movement, okay, it is flip flop movement, it is present only for lipids, not for proteins. What's the reason? Can anyone tell? Why the flip flop movement is there only for the lipids and not the proteins? Can you tell? No, no, that is not the case. You have to simply think if there is a protein attached to it, okay? So that protein, it cannot move here and it cannot exchange this lipid then. Why that is so? The protein cannot do it. Why that is so? You have to think in terms of charges. The nature of the membrane, the charges, in those terms you have to think. Why it is not possible for the protein to do flip-flop movement?
So it cannot travel across the plasma membrane because materials are there.
Correct. Very good. Very good. ताली बजा दो भाई. ठीक है. So since these are charged, okay, so these can basically this part is hydrophobic. So if this is hydrophobic, the movement, that is the flip-flop movement of the protein from outer to inner or inner to outer leaflet is not possible because it has to traverse through the hydrophobic membrane. Okay, this is the basis of the transport across the membrane and we will see how this is useful. So now first we have understood about all these things, right? So now the another reason for asymmetry of the membrane. As I told, the membrane is asymmetric, right? So one reason was the difference in the composition of different lipids in the membrane. Another reason which is true, which is there, is the presence of these carbohydrate moieties. Okay, so what happens is on the outer leaflet you will have a lot of proteins which are there, which are attached to the membrane. Either it can be glyco proteins, okay, it can be glycolipids, and basically why they are present, can anyone tell? Why they are more on the outer leaflet? Not for transport. Any other reason you can think of?
So they're exposed to the environment.
Yeah, they are exposed to the environment. Why? There would be a reason, right? Yeah, correct. So basically a lot of hormones which have to carry out the cell signaling. So these glycoproteins or glycolipids basically they act as receptors, okay, for cell signaling. So what happens is, let's say any of the you can say let's say any of the protein is there which is coming into the bloodstream, okay, and it is interacting with these receptors. Then what will happen? The conformation change happen. The signal goes to the inside, and a lot of events are happening inside. A lot of reactions happen inside because of the signaling. So for the signaling purpose, these glycoproteins and glycolipids they are basically present. Okay, and they're more on the outer leaflet. That's why this membrane is, again we call it as a asymmetric membrane, because on the outer leaflet you have a variety of glycolipids, glycoproteins which are present, and on the inner leaflet you don't have any of those. Not any, but they can be some, but not as much as compared to this, because this is exposed to the surrounding, right? So it is there so that the surrounding molecules can interact with it and carry out the signal towards the inside. Okay, is that clear to everyone? Right? So we talk about cholesterol. One important factor about cholesterol is it is only found in animal cells. Okay, and in other organisms you will not have cholesterol, but you will have different membrane stabilizers, which can be ergosterol or stigmasterol. So these are different variations of that structure, but not specifically cholesterol. Cholesterol is found only in animal cell. This is again a question that can be asked for your mostly your NEET exam. Okay, so I hope this part is clear, right? All the things are clear in this, the movement, the asymmetry, and the type of proteins you have. Is this clear? No, other proteins, so they can move with it because of the lipids also. Let's say if there is a protein, this is a lipid. If the lipid has moved, there is a space that is created. So now the protein can also move with that flow, right? That is also possible. Clear? So now there is one thing which comes, that how can you how you can isolate membrane proteins. Okay, so how you can isolate membrane proteins? So you have two types of proteins, right? One is peripheral, another one is your integral proteins, right? And in the integral proteins you again had two types. One was a transmembrane, that is fully embedded, and one can be the partially embedded. Now when I say that a protein is embedded fully across the membrane, right? A protein is present fully across the membrane. That means the protein, this part, it will be non-polar, right? And for this part of protein to be non-polar, there has to be non-polar amino acids which are there. For example, leucine, isoleucine, valine, right? All these non-polar amino acids will be there in this part. Okay, that's why it forms a transmembrane domain. Now, how will you isolate the membrane proteins? Can anyone tell? How you can isolate them? So these are the questions which are of the thinking base. Everyone will study about cell membrane, the proteins, but let's say if you want to actually study them, how will you isolate them from the membrane? In the integral, as I told you, there are of two types, right? One is a transmembrane, that means that is present across the membrane. Okay, it is embedded fully in the membrane. Another is a partially embedded, which is present only in one of the leaflet, either outer or inner, like that. Okay, so now how will you isolate them? Any idea? How can you think of isolating the proteins? So if you think about the peripheral proteins, if this is the membrane, this is the group, this is the protein. So the protein is interacting with the lipid head group, okay? And the protein will have some charge, right? And this is interacting with it. So for this case, you use chelating agents and salts, okay? Chelating agents, for example, EDTA. Have you heard about EDTA? What is EDTA? So what does it does? What it does? So basically, this has these acetate groups which can chelate a positive charge ion, right? So it can just surround that particular ion, and after surrounding, it can just help in taking it out of the solution, because this is surrounded by these basically molecules, right? So similarly, for this case, what happens is this chelating agent, it will just surround this protein, okay? And after surrounding the protein, it will bring it out of the solution. It will help in breaking this interaction, because this interaction is a weak interaction, okay? So this interaction can be broken very easily with the help of chelating agents or some salts in high concentration, which will surround this protein and will help in keeping it out of the solution. Okay, so this is how you isolate peripheral membrane proteins. Now if you think about these proteins which are integral, transmembrane or partially embedded, okay, this is fully embedded and the one can be partially embedded. So how will you isolate these? For these, you need something which can interact both with the non-polar and the polar part. Okay, so that is something you call as the detergent. Okay, so you can use detergents. Okay, so now here you also have two types of detergents. One can be non-ionic, another can be ionic detergent. Okay, so ionic detergent example is SDS. Have you heard about SDS? Sodium dodecyl sulfate. Okay, so SDS is a detergent in which you have basically long carbon chain and you have this sulfate group. Okay, so because of this part becomes polar, this part becomes non-polar. Okay, now what is the use of this? What will happen is this part which is exposed, this is hydrophilic, so this can be there with this part, this can interact with the polar part, and the non-polar tail, it can interact with this part, right? So overall this will surround this whole protein and it will help in isolating the protein from the membrane, because this has both the components, polar as well as non-polar, because this is a ionic detergent. Now if we talk about a non-ionic detergent, example is Triton X100. Okay, so this is used to isolate the membrane proteins in their native form. But if you will use SDS, you will denature the protein. Okay. Now, how does it happens? So basically, if you talk about a non-ionic detergent, okay, that is Triton X100, okay, what it does is, since it is non-ionic, so if this is your protein, right, and if the detergent you are using, it is non-ionic, right, so what it does is it can interact the hydrophobic part of the detergent, it will interact with this, it will surround this protein. Okay, and it will simply so this part will be polar of the detergent, but it is non-ionic, so it doesn't have any charge, but it can have polarity, right, it can be polar. So now this will just surround the protein, correct, and this will help in isolating the protein in the native form. That means the conformation of protein will not be hindered, because this has only interacted with the non-polar part of the protein. But if you are using a ionic detergent, for example, if you are using SDS, what SDS does is that since it has this polar part that is negatively charged, and it also has the non-polar part. So SDS interacts with both the domains of the protein, polar or non-polar. Okay, it completely surrounds the protein, and in this process, this is a harsh detergent. So what it does is it denatures the protein. It changes the structure of the protein. So overall, your protein integrity and folding will be compromised when you use ionic detergent. So the best thing you can do is always use non-ionic detergent for isolating integral membrane proteins, for isolating peripheral membrane proteins which are on the periphery, which are under weak interactions. You can just disrupt these interactions with the help of chelating agents or salts. Is this clear to everyone? Any doubt in this part?
Just forward 1 second. Just forward 1 second. Just put over it up.
Okay, okay, sure. So basically, as I told you, you have two types of proteins. One is peripheral protein and another is integral or transmembrane protein, right? The peripheral proteins, they are under weak interactions with the lipid head groups. Okay. So these weak interactions, they can be broken, and to isolate these proteins, you can use chelating agents or salts. These chelating agents or salts, what they do is they surround the protein because of the charges, and they just help in breaking these interactions so that the protein is isolated from the membrane. Okay, since these are weak interactions. Now for integral proteins which are either fully embedded in the membrane or they are partially embedded. Partially embedded means within a leaflet. Fully embedded means across both the leaflets, inner and outer. So you will have basically the inside the membrane, the core, the non-polar part is there. Outside you have the polar part. So you can have two detergents, right? One detergent is non-ionic, another is ionic. Non-ionic example I have given Triton X100. It will help you to isolate the protein in its native form. Native form means that the protein is preserved. The protein activity, the protein structure has not been disrupted. But the ionic one, that is SDS, sodium dodecyl sulfate, this if you use, it will denature the protein because it is ionic. So it will interact with both the domains of the protein, and there is a chance that while isolating this interaction, it can lead to basically disrupting the folding of the protein, and it will denature the protein. Is this clear to everyone now? So after this, we have the transport across the membrane. But before that, we will see one thing. So there are four types of membranes. Okay, there are four types of membranes. One is, you can call as permeable. Another is semi-permeable. Another is selectively permeable. The fourth one is impermeable. So, do you know the meaning of these terms? Okay, please tell.
From table through which the plant can like pass through it easily. Variable some things can things pass through it, but some cannot. And selectively variable is doesn't mean that only selective item can like pass through it, and for if you are permeable, no item can like pass through.
Correct. So basically, permeable, it means that something which allows everyone to pass. Okay, which allows everyone to pass through it. Okay, so that means that both solutes as well as solvents can pass through the permeable membrane. Any example of permeable membrane, if anyone knows? Any example can you recall? Which allows everything to pass through it. Cell wall, very good. Clapping. So example is cell wall, right? Now semi-permeable. So the word semi, it means half. Okay. So that means it allows solvents, but it not allows the solute. Okay. So the example can be the egg cell membrane that you see. Okay. So that is semi-permeable. Okay. The third is selectively permeable. Selectively permeable is something which allows selective molecules to pass through it. And impermeable means which do not allow anything to pass. Now, one question is, the cell membrane or your plasma membrane, is it semi-permeable or selectively permeable? Selectively permeable. Many people have this doubt. They mark it as semi-permeable with confusion. But the cell membrane, it is selectively permeable. Okay. So example is cell membrane. Right. And now impermeable. Impermeable is something which does not allow anything to pass. Can you give an example of impermeable? Can you think of it? So have you heard about cuticle, which is present in plants? So what happens is, in plant cells, in many of the cells, there is a deposition of some substances across the cell wall, right? For example, lignin, In case of cuticle, it is the waxy layer that is formed. But yeah, there are these lignin depositions and all this, which makes the membrane impervious. Impervious means the same, that it doesn't allow any molecule, whether it is solute or solvent, to pass through it. So is the 4 types of membrane clear to each one of you, right? You got it? So if you understand this, now we will understand, since the cell membrane is selectively permeable, we will understand the transport across the cell membrane. So first one is the passive transport. Okay, first one is the passive transport. What do you mean by passive transport? Which do not require energy, which can happen on its own. For example, if you reach to the top of a mountain, you drop a ball, that will fall by gravity, right, on its own, right? So something like this. So that we also called as downhill movement, okay, which can happen on its own. And why it is happening on its own? Okay, so what happens is there is something called as a concentration gradient. Okay, so let's say if here there is a source of particular gas or a perfume, if I leave it here, it will diffuse to every corner of the room, right? So this is something that something moves from higher concentration to lower concentration. So this is how the passive transport works on. So it does not requires any energy or ATP. Molecules move from their higher concentration to lower concentration, that is down the concentration gradient. Now these can be of 2 types, okay? So if you understand about passive transport, one can be simple diffusion, okay, which mainly happens for gases. So for gases, what happens is that most of the gases which are like oxygen, okay, nitrogen and all the gases, they can simply pass through the membrane, okay, they can simply pass through the membrane. If you, let's say, talk about oxygen, okay, so is oxygen polar or non-polar? Non-polar, right? Because if you see oxygen, both the dipoles, they so basically first thing is that there is no electronegativity difference, right? So these gases which are there, O2, N2, okay, CO2. So in case of CO2 also, you will see all the gases, they are non-polar, they can just pass through the membrane, across the membrane, right? Now the similarly to gases, water can also flow, okay, but in case of water, what happens is this is polar, okay, water is polar, because if you see water, here you have the lone pair of the electrons, right? So since water is polar, in majority of the cases for flow of water, you have some aquaporins. If you have heard about these terms, these are some channels which help in the flow of water, okay. So the diffusion of water, we call it as osmosis, okay. Osmosis is the diffusion of water from higher to lower concentration, but for gases we have the term as diffusion, okay. So neutral molecules which are non-polar, they can simply pass through the membrane. Is this part clear to everyone? Okay, now there is something called as a facilitated diffusion. Facilitated diffusion is for mostly your polar molecules. Polar molecules, okay. So what happens is for the polar molecules, since they have a charge, to transport them across this membrane through this hydrophobic core, you require some channels or some carrier proteins which can help in this particular process, okay. For example, if you have to transport glycerol, okay, so we saw the structure of glycerol, it has 3 OH groups, right? So it has some polarity. For example, if you have to transport glucose, okay, so glucose also has OH groups, right? So because of this, if you want to move the molecules from the outside to the inside, you require some carrier proteins. So that transport which requires a carrier protein, that will be called as facilitated diffusion, because it requires someone to facilitate the movement of molecules from outside to inside. Is this clear? Now the third one that you can call is your active transport, okay. So active transport, what does it mean? Which requires energy, right? So it's something like if you want to transport something from the down of the hill to the top, so you require energy, okay. So if you live in mountains, for example, let's say, have you eaten Maggie in mountains, right? So for them, if you will talk to the person who is selling it, he will say that the Maggi is of very high price. Why? Because we have to transport from the bottom to the top, right? So there are transportation charges. So it requires energy, okay? And for the cell, the energy requirement is in terms of ATP. So ATPs are required, okay? And in this case, you have the pumps, okay, which are needed to move the molecules against their concentration gradient. That is the movement which happens from lower concentration to higher concentration. If you want to move those molecules, you require to spend energy, okay? And basically this requires some pumps. So the pumps can be sodium potassium pump, sodium potassium ATPases. So these are required for, let's say, the transport of sodium and potassium ions across the cell membrane. Now this active transport, it can be of two types. One can be primary active transport, another can be secondary active transport. In case of primary active transport, you transport only one molecule, right, from the lower concentration to higher concentration. But in case of secondary active transport, you are transporting two molecules. So this is something that you called as co-transport. Co-transport means that the two molecules, either they can move in the same direction or they can move in the opposite direction across the membrane. So these are of two types. One is symport and another one, can anyone guess? Antiport. Very good. So symport, in this what happens is, both the ions that are there, okay, and two ions, they simply move across the membrane. For example, this is for sodium glucose. So glucose, if it has to move across the membrane, it requires GLUT proteins, which are, for example, GLUT4. Okay, so these help in the transport of glucose across the membrane. And this is symport because the transport is happening in the same direction. But if you consider antiport, in case of antiport, what happens is, if one ion is coming in from outside, the other ion, it simply goes out from inside. So this is happening in opposite direction, okay? And this is for sodium and calcium ions, which is present in heart. So in heart cells, mostly antiport happens to maintain the level of calcium and the contractions and everything. So basically, these are two type of active transport. Is this part clear to everyone? Clear? Now, any doubt? Okay, so now we will start with some questions. So you will just answer what will be the option. Okay. First question is, plasma membrane is selectively permeable. Correct. We have seen this already. Then the most abundant lipid in the cell membrane. Which one? Everyone say. Option D. Phosphoglycerides, right? And among phosphoglycerides, which is most abundant? Lecithin. What it is called apart from this? PC. What is PC? Phospho. What was C for? Choline. Phosphatidyl choline. Very good. Integral cell membrane proteins are. Read all the options. Don't just solve in a hurry. All of these? Other people? All of these? All of these? So partially embedded in the membrane, we have seen that for the membrane, these can be present in either one of the leaflet, or they can just span the membrane because of the non-polar amino acids which can be there in the proteins. Show lateral, okay, they can show lateral movement, but not vertical movement, okay, that is flip-flop movement. So this movement is absent. Flip-flop movement is not there, but lateral movement is there within the lipid bilayer. So the answer becomes all of this. The partially embedded. So the partially embedded proteins mostly they can act as a site for interaction for signaling molecules, again because they are present on the periphery, right? And since they are also interacting with the lipids, okay, and they are embedded in the membrane, so some signals which can like be there for the cell to divide, or on those cases these can communicate the signals to the lipids also, so that the fluidity of membrane can change accordingly. So these are some of the functions. Now, the lipids are arranged within the cell membrane with C option, head towards the outer side and tail towards the inner side that we have seen. So, if this is the whole membrane, okay, and if you see, basically, if you talk about any one of the leaflet, right, in this case, so the head is towards the outer, the tail is towards the inner. So, C option. Now, this is a question in which came in next exam, okay. So this is about the transport of molecules. What will be the order of transport of different molecules? So the permeability of molecules across a membrane depends on their size as well as the solubility, right? So these are the two factors you have to think of in a non-polar solvent relative to their solubility in water. For the species oxygen, water, potassium, glycerol and glucose, the order of their permeability across lipid bilayer in absent of any protein transporter is. So there is no protein transporter which will help in or which will facilitate the movement. It is on their own. Okay. On their own, what will be the order? So can you think of this question? So there are two factors if you think of. One is size, another is solubility. So whenever you read such a question, what should be the approach to solve the question, right? So the approach should be you are given the factors in the question itself. In NEST, what happens is most of the questions, the theory is already given in the question. You just have to deduce the answer, right? So now you have two factors, size and solubility. You have to think if one is more, what will be the outcome? So if you guys think if the size is more, okay, then will the transport be faster or slower? Slower, because a molecule with a large size, okay, some person who is very fat, he will run very slow, right? So that means that here the transport will be slower. And this will be vice versa. If the size is less, then the transport will be fast. Okay, now talking about solubility. Okay, solubility simply means the polarity. Something is soluble in a solvent if it is of the same polarity, right? So if we talk about polarity for the non-polar molecules, will the movement be faster or slower? Slower, why? So no, don't think of attraction. You think of moving through the membrane. So if you are moving through the membrane, you have to cross this, right? So here, what is this environment? Non-polar. So if the molecule is non-polar, it can move easily through the membrane, right? So if the molecule is non-polar, will the transport be faster or slower? Faster. So the transport will be faster. Now, same, this will be the vice versa. If the molecule is polar, the transport will be slower. Now, can you tell the order based on this information? You have to think of all the molecules. So what happens is in the plasma membrane, you have the lipids, right? So this tail forms the hydrophobic core. So what happens is if the molecule is polar, that means if the molecule has some charge, right? So this charge, so basically this doesn't want the charge species to be here. This wants the non-polar part only. So what happens is for the transport of charge species, this either the transport will not happen or the transport will be very, very slow. Okay. And in case of non-polar molecules, since they don't have any charge, they can easily interact with this and they can move very easily through the membrane. Got it? So now, can anyone tell the answer? Option A. How many people say option A? Only one hand, two hands, three, four, five. Okay. So let's think in this way. Okay, so you have oxygen. Is oxygen polar or non-polar? Non-polar. And it is non-polar and it is a gas, right? So this can move by simple diffusion very easily, correct? So oxygen, it will travel the fastest. That is clear from this. Now the second thing you can think of is water. Water, if you talk about, water is polar, but this is uncharged, right? So the movement of water will be after the rate of movement will be the after the oxygen movement, right? So this will be water. Now you have glycerol and glucose. So let's understand about this. So if you talk about glycerol and if you talk about glucose, so I am writing some factors, the size, which has more size, glucose or glycerol? Glucose, okay, the size is more, here the size is less. Now the other factor we have to think about is polarity, right? According to the question. So polarity. So which has more polarity, glucose or glycerol?
So we can't reduce that directly.
What?
The glucose structure is like a ring,
Correct.
and there is one CH2 OH group on the top.
Correct,
Which is called
correct.
high polarity.
Ah.
There's one O in the middle that is also causing a polarity difference. So the vectors will act in a very weird way.
But
Glycerol, the vectors
yeah,
act directly.
correct. But if you are thinking about the net molecule, you have to think in terms of solution. So if a glycerol is present in water, it is surrounded by water molecules, right? So you have to think about the interaction. In the ring also, if you see the ring, in the ring also here you have the OH groups, right? And here also you have the OH group. So if you think the number of OH groups in case of glycerol, how many OH groups were there? 3 groups. In case of glucose, the OH groups are how many? 3, 5 groups, right? You have seen the structure, right? This is a aldehyde. So on the top you have a CHO, then like this, right? So this carries out till 6 carbons and you have these groups. Okay, so overall the polarity, if you say, for in comparison to glycerol I am talking about, it is relative polarity. So here the polarity is more and here the polarity is less. Clear? Clear? So now which will move faster? Glycerol will move faster because the size is less, so it can move easily. The polarity is also less, so it can move to across the membrane, right? So in this case, the second should be glycerol and the third should be glucose. Now, can anyone talk about the potassium ion? What will be the case for potassium ion? So first thing is, so as he was saying, potassium has a positive charge. So this is polar, right? One criteria is there, it is polar and it is charged also. So the polarity is very high, correct? Now you think in terms of size. What do you think? The size of potassium is more or it is less? It's small. Why you think it's small? If you think in terms of solution, the size will be more because what happens is potassium, it will be surrounded by the water molecules, and because of the hydration by these water molecules, there will be effective increase in the size of the whole molecule, right? So because of which what happens is the effective size of potassium ion in water, if it has to travel through this membrane, okay, this size is also very high. Okay, so size is also high. The size is high and the polarity is also high. So both the factors are high in this case of potassium ion. Is this clear to everyone? So overall, if you now see the order of transport, you will have potassium at the last. So the correct answer is option A. So is this logic clear to everyone? How you can deduce the answer with the given information? So whenever you get a question in next exam, you have to think about the things which are given in the question. You have to think how, if you modulate that particular parameter, what effect it will have, what will be the outcome, and then based on the given species, how that parameter will vary. So this is how you have to think and come to the answer. Is this clear to everyone? Right? So what's the time? 1:20. Okay, let's solve this last question and we will end the class. So, okay, okay, we will solve it in next class. Fine.